the way that E. coli, at least, or many bacteria, responded naturally through evolution to aldehydes is to detoxify them. And so it's hard to impart an intent really to microbes, but given that aldehydes, particularly of shorter chain length or the kind of aldehydes that you might get of medium chain length that could come from an error in fatty acid biosynthesis, a stress response of some kind. There are some, but maybe not that many different kinds of aldehydes that natural metabolism might generate. Predominant aldehydes being something like glyoxyl, an overflow metabolite from glycolysis, central metabolism. And when you get those aldehydes, you can have a cascade of negative processes occur in the cell. So what we knew back in the early 2010s was that there are many different kinds of enzymes that seem like they would act on not just glyoxyl, but a few different kind of aldehydes. And if you added any of these aldehydes to cells, you would rather quickly, meaning on the order of 30 minutes, an hour, see conversion of the aldehyde quantitatively to its alcohol form. You could also, if you were looking at transcript levels, you'd see some kind of set of genes, often encoding candidate enzymes, be upregulated. You could purify some of the enzymes and then test them in vitro with different aldehyde substrates and try to figure out what they do. But based on sequence similarity, you would look at the genome, say even of E. coli, and there would be potentially 20 or 30 of these. And so that seemed like a problem that may or may not be possible to overcome. Essentially making the conventional notion in the field be that aldehydes are not something that E. coli could overproduce. And so this just kind of represents the problem as, shown here, 6, but that's not very precise enzymes that might be in the genome that do this that you don't want. And so I got started, initially on a slightly different project, trying to think about the aldehyde as a potentially toxic intermediate, and something that you wouldn't want, but that maybe the recruitment of enzymes that would make it and then use it to do something else. If you could recruit those enzymes close to one another, then you could have an aldehyde almost shuttled or at least not freely diffused throughout the cell. This kind of notion is called substrate channeling. And you see that actually occur in some naturally evolved systems. But as we spent more time looking at model pathways and literature, well, for one, it seemed difficult to construct a model pathway where you would both make an aldehyde and then use it to do something else if there were so many other enzymes in the cell that were just converting it to an alcohol. And so it was in the spirit of trying to solve that problem, as well as recognizing that, in a lot of these cases, that aldehyde is worth more, both monetarily and also more useful than whatever next molecule we might try to make from it, that we got started on some exploratory knockouts. And so we've talked about genome engineering and how to do knockouts. We attempted knockouts of candidate enzymes. And if you look at the E. coli genome as represented by this circle here, there were really 2 categories of enzymes that we were focusing on the most, 2 superfamilies of enzymes. And I'll get to introducing those in a little bit more detail in a second. The way that we would then actually try to investigate this question is we had just stumbled upon and started to do some characterization on an enzyme, a carboxylic acid reductase, also known as a CAR, from a bacterial species that could convert several different kinds of aromatic carboxylic acids, whether it's benzoate shown here in black or vanillate shown here with the blue ring substitutions. Carboxylic acid reductase, and you saw this in your homework, can, in the presence of NADPH and ATP, perform this kind of reduction to get the molecule that we wanted to look at, the actual aldehyde, made in the cell. And part of the reason why we liked this approach was because from an experimental standpoint, we didn't want to douse the cells with aldehyde, and we weren't sure if the aldehyde would go into the cell. We were doing some what are essentially called metabolite supplementation experiments, and we weren't using common sugars. Thinking back to that bow tie picture of metabolism, we don't really know that these are going to get how they're going to participate in metabolism, and we actually don't want them to participate in metabolism, right? We wanted to have this sort of thinking about an orthogonal metabolic pathway, something that we can add and we can monitor and we can close the mass balance, so that we're not like in the case of a 13 carbon labeled isotope, that kind of molecule. We don't want to trace like where the flux is going through the cell. And we knew, and we could empirically verify that we got really good uptake of just applying these simple aromatic carboxylic acids. The way we could tell that is, if we transformed our E. coli to express this kind of CAR, we pretty readily saw formation of the aldehyde. But, what we would actually see is very little accumulation of this and just having it go all the way to the alcohol. And so what we then tried to essentially do is to combine these, to make these knockout strains, to transform them with CAR, to supply this carboxylic acid, and then see what happens. So in terms of trying to do this in a rational way, we looked at the literature. The literature really focused on these two superfamilies shown in this blue color coding, are the aldo-keto reductases, or the AKRs. And there was some in vitro data on, in particular, these three enzymes, which seemed like relevant candidates. Remember, our objective was just to accumulate aromatic aldehydes, and we focused on benzaldehyde as a model substrate for that because it's relatively simple. And so we think, we sort of thought if there's an enzyme that's gonna take benzoate and make benzaldehyde, that's gonna take benzaldehyde and make benzyl alcohol, then it would probably take something like vanillin and make vanilla alcohol. And you can see the difference in chemical structure between these substrates that were tested, where benzaldehyde, even in these studies, is sort of a reference that's not physiologically relevant, whereas methyl glyoxyl and phenyl glyoxyl are actually generated by the cell. And when you look at the specific activities in these columns, you can't, again, draw too many inferences here about what nature evolved these enzymes to actually do. But when you see this kind of order of magnitude increase in activity on a substrate, you can say it's the preferred substrate. Back in the day, and particularly if you have a collection of natural metabolites that are all present within the cell, and at what relative concentration they are, you can do this kind of study, provide these substrates at those concentrations, and then you can say, based on what might have the lowest Km value, you could say, oh, well, this is actually the substrate that this enzyme is designed to react on. It gets a little hairy when you're talking about something that the cell doesn't ordinarily see. But the point here is really that while the activity on benzaldehyde was considerably lower than some of these other substrates, it's still, and it's hard to say this objectively, but these are still high, relatively speaking. And so we wanted to target, in particular, these two. You can see a bit of a drop off here, but YEA-E or YEA-E was also on our short list. And so as we were making this short list, thinking again about the tools that were available to us, and actually for this work, I knew about but didn't choose to use a multiplex automated or automatable genome engineering strategy. And the short reason for that was because the method was still pretty new at the time. And there was, from sources that I had discussed with, more certainty around doing things in the more manual and sequential way. Definite trade-offs associated with that. Sometimes being an early adopter is a really good thing. Other times, early adoption can be a pitfall. You don't necessarily know. I mean, in general, what you're trying to investigate is not how to make these mutations. It's what the knockout actually does. If you're in grad school and you're faced with an opportunity to use a really new and powerful method, but you're not certain about it, it's not an obvious choice what you should do if there's like a very well characterized but slower way to do this. So in the case of the methods that we were using, it seemed reasonable to be able to make about 5 to 10 knockouts. Going beyond that would seemingly take too long. If we look at the other superfamily of interest, these alcohol dehydrogenases, which also convert your aldehydes into alcohols, there were these enzymes, YJGB and YAK, reported in the literature to have activity on these substrates. And now you see a different set of substrates. So alcohol dehydrogenases natively, the most common are involved in the conversion of acetate to acetaldehyde to ethanol. Some are actually dually functional. But as ethanol is a major microbial fermentation product, alcohol dehydrogenases were relatively well characterized on those kind of substrates. The question then is how many of those alcohol dehydrogenases might have cross-activity with something like benzaldehyde or hexanal. And you can see actually here, in terms of catalytic efficiency, these alcohol dehydrogenases actually have greater activity on benzaldehyde and hexanal than they did on acetaldehyde. And there was another one that was really well documented in the literature too, but it didn't have catalytic efficiency documented on these particular substrates, so I didn't include it on the table, but that's YQHD. And so maybe one thing to pause and just reflect about here is that this is pretty unusual, that you would have, E. coli has all of these enzymes. They are all acting on multiple different substrates that are pretty structurally diverse. If you just look at the left-hand side here, there's a lot of diversity in these structures. That means that these enzymes are really just looking for the aldehyde, and it almost doesn't matter what's on the rest of it. Physically, when we were talking about protein engineering from a structural biochemistry standpoint, that suggests that there's some kind of pocket that might be large, where the aldehyde enters the binding pocket, and maybe this binding pocket is large, you could say, or shallow but exposed in a way that allows it to, if close to the surface, not be so dependent on binding to the rest of the chemical structure. And so it seemed early on like these genes, if knocked out, might have an influence on not just one aldehyde substrate, but potentially many. And I will talk about that momentarily.